This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 339, recorded on May 29th, 2015. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
Missed you for a couple of weeks.
Yeah.
Do you miss the TWiV experience?
I miss the TWiV experience, and it's not bad to listen to. You know, I have to say, I do listen to the shows that I'm not on.
I don't, because I'm not on none of them.
Right.
I was not on one once.
Right. I remember that.
And I listened to that one. That was fun. Also joining us from southeastern Michigan, Kathy Spindler.
Hi. I just realized I spoke before I was introduced again.
It's
Sorry.
Not a problem.
Yeah.
Kathy and I were in California last week. We had a good time. It was a good trip, right?
Yeah. Nice place.
And we had a big audience. It was fun.
It was a huge audience, yeah.
And I love the paper. I thought that was pretty cool.
Mm-hmm.
Yeah.
We actually have a follow-up from Alan Dove about
yes, you do.
that.
Yes, so I was talking out loud to you, but you couldn't hear me.
Yeah.
So you asked Coleoptera, those are the beetles.
Right.
And Hymenoptera is wasps, bees, and ants.
Right. And I realized that after I said that, what it was about the phylogenetic tree that I recognized is individual insects like the housefly, the red flower beetle, fire ant, and silkworm. But I was not really up on the orders as well as Alan would be. So,
have another follow-up from last week's episode. E.T. writes, "Hi TWiV folks, I'm having a blast listening to this podcast. Coyotes have sushi. Wanted to drop you a quick factoid, re mosquitoes. It may interest you to know that the deadliest organism on planet Earth is the mosquito. These creatures can kill something up to 5,000 times its size." From my handy book, The Biology of Science Fiction Cinema, Mark Glassy.
I would disagree with that because a virus can kill something millions of times its size.
Absolutely right.
And even if you argue that that's not an organism, a bacterium can kill something millions of times its size.
I mean, deadliest really also has a frequency involved with it, right?
Yes, and certainly by that standard, mosquitoes would be in the running.
But mosquitoes are not actually the killers. It's what they inject, right?
It's what they carry, yes.
So I'm not so sure. But anyway, we appreciate the thought.
And thanks for phoning home, E.T. Or is that Ed?
I don't know.
E.T. or Ed.
Could be
Vincent, you can refer people who send you their screenplays or fiction outlines, whatever. You can refer them to this book, Biology of Science Fiction Cinema.
I have to look at it.
Yeah.
This 2001. It could be slightly outdated, right?
Yeah.
Then we have follow-up for the previous episode, Steamer. Paul writes, to the TWivers, really enjoyed episode 337, Steamer. Follow-up question, isn't a tumor or cancer by definition derived from local cells? So wouldn't hemocytic leukemia be classified as an infection? Unseasonably cool 14C with light showers falling to alleviate the local pollen alert in Philadelphia. I asked Steve Goff, and he said, offhand, I'd say I never heard of that definition of a tumor or cancer. I don't think it has to be local, does it? It's just uncontrolled growth of cells, right?
Yeah.
Right, because then anything metastatic would not be cancer.
Yeah. All right, we also had a kind of continuing discussion about, all right, remember the paper on patient P? You guys, of course, remember.
Mm-hmm.
This, and for those of you who don't remember who are listening, this was a patient who had a couple of mutations in the gene encoding IRF7 that predisposed the patient to serious influenza. And at some point, someone asked about the role of these mutations in leukemias and cancers. So I started emailing people. We got a couple of emails about this. We got one which we read, I think, at San Bernardino, of a theory. So I emailed the author of the paper, Jean-Laurent Casanova, and I said, we did your paper on TWiV. And he wrote, many warm thanks for your interest in our little story. And I have to pause and say, so he thinks this is a little story.
A little story.
And we were commenting how much data were in that.
I'd hate to see what he calls a major work. Wow.
I don't think we can say that germline IRF7 mutations predisposed to leukemia. Homozygous and heterozygous individuals with known variations in IRF7, including the family we recently reported, do not seem to be prone to leukemia. As per somatic mutations in IRF7, I'm not familiar with the literature. They might indeed be drivers of passengers of certain malignancies, including leukemia. Again, I'm not familiar with these studies. And then the first author on that paper, Michael Ciancanelli, wrote, I was flattered to hear you and the TWiV team cover our paper. Your discussion was fantastic. Many, many thanks. I think the reference to leukemia in your podcast was to other research that defined a role for IRF7. Our work obviously focused on the involvement of IRF7 in immunity to infectious disease, but I have found the papers that refer to the anti-tumor effects of IRF7. To our knowledge, our patient P has not developed any malignancies. However, in light of IRF7 suppression in several cancer models and tumor regression upon restoration of IRF7 expression or treatment with exogenous interferons, she may be at an increased risk for some cancers. Additionally, there is the connection of IRF7 with Epstein-Barr virus infected cell latency. Here it appears that IRF7 is induced and activated by one of the viral proteins, LMP1, and this could drive the establishment of latency. I'm unsure what would be the effect of the loss of function mutations identified in our influenza patient, but again, something that her physicians should monitor as she is seronegative for EBV. But none of this gets at the question of why would tumors be defective in innate immune pathways, right?
Mm-mm.
Right.
We don't have an answer. So, thank you for your follows up.
I mean, I could come up with a teleological answer that helps the tumor evade immune surveillance, but how it arises, I don't know.
Yeah, so the person last week suggested that because tumor cells are dividing quickly, there must be a lot of DNA and RNA in the cytoplasm, and that's stimulating the innate response, so therefore tumors that downregulate that, they survive.
Those are the ones that survive, yeah.
But I don't know that there is DNA and RNA any more than normally in the cytoplasm of a tumor cell. I mean, that's speculative, I think. Anyway, a couple of snippets sent in from listeners. This week we had a death here in New Jersey caused by Lassa fever virus. This was an individual, and Justin sent us the CDC release. There's also an article in the Times. Lassa fever in a traveler returning from Liberia. He came to JFK, was fine. Then on the 17th of May, he arrived in the U.S. On the 18th, he went to a hospital in New Jersey, which is always a bad idea.
We're going to get letters about that.
Hey, but I live in New Jersey. And we had a doc on from New Jersey. Remember the guy from Camden? I don't know if you guys remember that. It was Virology 911, a long time ago. So on the 18th, he was asked for his travel history, and he did not indicate travel to West Africa. He basically lied, or he just omitted information, right? He was sent home, and then on the 21st came back with worse symptoms, was isolated. He was basically diagnosed with Lassa and died. So Lassa fever is quite common in Western Africa. It's occasionally exported elsewhere, but when it is exported, there's not person-to-person transmission typically. This is the 6th time Lassa has come to the U.S. since 1969. And in Africa, I think we talked about this recently when we did a Lassa paper, 100 to 300,000 cases a year and 5,000 deaths. The case fatality rate is 1%.
Right.
In Africa, you acquire the infection from rodents through contact with urine or their droppings, and it can go from person to person by direct contact with a sick person's blood or body fluids, mucous membranes, or sex. Sounds a lot like Ebola, doesn't it?
Mm-hmm.
Sure.
Not transmitted through casual contact, and people are not infectious before the onset of symptoms, also similar to Ebola. Well, there you go. It's interesting. This will happen often, right? More and more. I mean, travel and all that.
Mm-hmm.
We have to have scanners in the airports that pick up all your viruses as you walk through them. Probably you wouldn't like that, Alan, right?
No, it'd be kind of intrusive.
To know what your viruses are?
Yeah,
Oh, look,
getting a regular update on that. I don't know.
I want a daily update when I get up in the morning. I want a scanner in my house.
Good morning. Your viruses today are...
Yeah, exactly.
It could happen someday, but maybe not in our lifetime.
Yeah, probably not. Gosh, I wish I could live forever. I want to see all these things. You know, a BBC reporter called me this morning, and it's a really interesting question. He said, okay, someday we're going to be able to eradicate many infectious diseases. Right, and he said, yeah, probably most of them in the long term. And he said, and that's great because then people will be able to get educations and they'll have jobs. He said, but aren't we going to be overpopulated? There's not going to be jobs and food for everyone. And, you know, that's a really interesting way to look at it.
People have been making that argument for centuries.
Really?
That when medicine gets better, it will overpopulate the planet. And from the evidence I see, we're overpopulating the planet without medicine having to get better.
Yeah, that's what someone said on Twitter already.
Yeah, we're up to, 7 billion people now and are going to be soon, and we keep just kind of finding new places to stuff them. And a lot of the population growth is happening in places that have abominable medical care.
Well, you know, there is a prediction of plateauing at about 10 billion, right?
Right, and the idea, actually, there's usually what you see in development when public health improves is that population growth slows and then in most industrialized countries actually reverses.
Yeah, right.
So if anything, and the reason's very simple, if parents know that their kids are very likely to live, then they're only going to have a few of them. Whereas if you don't know how many of your kids are going to live, you're going to have a bunch of them to maybe have one survive to adulthood to help support you. So that's kind of the way things have developed classically, is that as more children survive, as any parent can tell you, children are very effective birth control. So people have fewer of them, and I don't think there's a danger of better health care or better public health increasing population. I think it's going to do the opposite.
You know, this doesn't happen overnight, as we see with the polio eradication program. So it's a long-term thing, and then I think we will figure out how to. I mean, you know, I told him, you know, we should be building vertical farms in countries that can't provide enough food, right? And that's partial.
But that's another. Providing enough food is not generally the problem. It's getting the food. The famines that you see occurring in the world are political. It's not that there isn't enough food in the world or that it can't be gotten to people. It's that whatever is going on in that location prevents food from getting to people. So it's so far we haven't maxed out anything as far as that goes. We've still got lots of stuff. It's too bad Dixon's not here. He would riff on this for hours. But there's still lots of stuff that we could optimize that would give us much more room.
You know, I told him that it's an interesting idea and he should write a science fiction book about it. And in the meantime, we have to really proceed to eradicate infectious diseases as best we can, because that's really one of the reasons we do all this research, you know, because we don't want. But the interesting question is, if you got rid of all the viruses in us or bacteria, we can't get rid of all the bacteria. If you eradicated all the viruses, say, that cause death in humans, I wonder if it would not be good for us. Right, it could be that some of them are actually beneficial, some of these infections, as we shall see.
The hygiene hypothesis.
In fact, we have a letter about that later on. All right. We've said enough so that we will get letters now.
Yeah.
Right. And I keep having to change the last 3 digits of the world population, but it's 7.3 billion currently.
7.3 billion, okay.
Yeah.
So it goes up 1, 3, 4, 7, 8, 9, 10. Oh, it's like many per second.
Yeah.
Wow.
And you can tell that the birth rate is faster than the death rate if you watch it long enough.
I don't know. It's just tough to bring people in the world now because things are getting tough. There are no jobs. All right. Let's be a little optimistic here. Let's see. I'm not sure who sent this in, but I have it here. It's a transgenic mouse model for a MERS coronavirus infection. And this is published in the Journal of Virology, which is not a journal we often talk about here on TWiV. Generation of transgenic mouse model of Middle East respiratory syndrome coronavirus infection and disease from University of Texas Medical Branch in Galveston. And basically they have taken the human dipeptidyl peptidase 4 gene, DPP4, which we talked about a long time ago, made a transgenic mouse, and they infect these with MERS coronavirus, and the virus replicates extensively in the lungs, also in the brains. And also they find RNA in other tissues as well, suggesting a disseminated viral infection. But it's a lethal infection. And
it's lethal with pneumonia.
Yeah, it's good. I don't know what the brain involvement means.
That's a bit of a red herring. And in fact, one of my students had proposed this in his final paper. And so I looked up what the status of it was, and I found this paper. And then I wrote to Matt Freeman. So anyway, I know that Matt has worked on another model that is not yet published, but I think will be presented at ASV, where they've made a different kind of a model with the DPP4 receptor for MERS under control of the endogenous locus.
So that could be more restricted replication as a consequence, right?
Right.
Anyway, so this is useful to study pathogenesis, antivirals, vaccines. And you know what's funny in this paper? They say at one point, after a great deal of work or something, we got these mice. Where is it? Go ahead, Kathy. With considerable effort, we succeeded in establishing a transgenic mouse. I rarely see that statement. A lot of things are...
a lot of work.
Yeah.
Go ahead. Sorry, Kathy.
Yeah. No, I was just going to say that I think that for a while, even this work was in the pause, even trying to make the transgenic mouse models. So that was then released. But then evidently, if you want to infect these mice with your virus, then that could be considered gain of function, possibly. And so you have to get a special approval for that kind of experiment.
It's crazy. This is really nonsense, this pause. Have I said that before?
I think so.
All right. We won't beat a dead mouse. Sandra, our friend in Texas, sent us an email, a link to a story about the first case of West Nile virus in Texas this year. Happens to be the neurovirulent form. In other words, you get encephalitis, which is not good. Texas reports first case, near the Gulf Coast. And there will be more, of course.
Yep.
The first one. But she says the good news is the only people in Texas diagnosed with chikungunya virus got it while traveling outside of the state. Yes. So chick is not yet, what would you say, autochthonous? Is that the right word?
Oh, I think that is the fancy word for it.
Or I would say endogenous, but it's not really the right one. Yeah, so people bring in chick quite often to the U.S., and there are a couple. I guess you put these in here, Kathy, right? Because it's purple.
Mm-hmm.
Yeah, autochthonous though would mean that it was indigenous, not that it didn't come from someplace else. And if chikungunya becomes established in the U.S., it still will have ultimately come from someplace else.
But like West Nile, we consider West Nile to be autochthonous now, right? Because it's established in the U.S., right?
Right.
Well, yeah, I guess based on the strict definition of this autochthonous word, though, I can see Alan's point. It's not native.
So, but nothing is native, right?
Nothing's native to anywhere, probably, except wherever it originated in the primordial soup. But I don't know. I would consider Lyme disease to be autochthonous. Mm-hmm. Right? That's been in the U.S. probably since forever. But something that came here within recent history. I don't know what the cutoff should be. Would probably be just naturalized, maybe.
Yeah, so indigenous rather than descended from elsewhere. Okay.
Yeah.
All right. Well, we'll see. Dixon would probably know, but I think that you get the point. The point is that West Nile is transmitted from person to person within the U.S., whereas chick they're all imported so far. I wonder if that will change.
It certainly could.
Yeah, why not? As long as the mosquito.
Right. Which mosquito species is chikungunya?
Well, it has recently adapted to Aedes albopictus, right?
Okay. Right.
I think previously Aedes aegypti is still the vector in many places, but it has an expanded host range because the virus is able to replicate better in albopictus, which is present throughout the U.S., as you know.
Yep, could certainly come here.
Being introduced, I think it came in the U.S. through Houston originally, or somewhere in Texas via the tire trade, the used tire trade.
Right, classic.
Our paper for today was suggested by Pete or Pyatt.
And Pete, I think.
Pete,
That's the Dutch way of doing Pete.
So the painter, Pyatt Mondrian. I mean, my mother always used to call him Pyatt, but what did she know? She was an English teacher. She's not
an American English teacher.
Yeah. So Pete, hi team. I recently found this article, which I thought was a rather interesting way that a virus manages to overwhelm a cell. And I looked at this, and this is a fabulous, fabulous article published in Nature Communications called Widespread Disruption of Host Transcription Termination in HSV, Herpes simplex virus type 1 infection. And I thought Condit would love this. He's not here.
Just to back on Nature Communications, is that the open access one?
Yeah,
Yes.
Yeah.
Okay, so everybody can take a look at this. Oh, yes, now I see open on the page.
You can read this paper. In fact, you can open it. I'll wait for a second while you get it and open it on your...
All the people who are out running with their iPods and listening to TWiV.
We will describe it so that you can understand it. The first author is Andrei Rutkowski, and the last author is Lars Dolken. And they are from University of Cambridge over in the UK, University of München in München, Germany, and also
Ludwig Maximilians Universität München.
Okay.
And then Christian Albrechts Universität Kiel. And Julius Maximilians Universität Würzburg.
And here's my favorite. Oh, it's the same university. Tennis Court Road.
Yeah, Tennis Court Road. I've been on Tennis Court Road.
Yeah.
Tennis Court Road because I thought it was such a cool name.
Did you get your picture taken there? You must have.
No, I didn't.
It's before cell phones, probably.
Yeah, it was.
Yeah, so Tennis Court Road, I remember from Watson and Crick days. Because you read their papers, Cambridge University, Tennis Court Road, Cambridge. It's so cool. And you think, these guys can go play tennis while they're on break. So the reason I thought this would appeal to Condit is because the story here is that herpes infection turns out to antagonize the termination of mRNAs. And that's what happens late in vaccinia virus infected cells, right? And that's why you get double-stranded RNAs made, which we talked about in that paper a while ago. But he's not here. Anyway,
it's also rather paradigm shifting, if true.
Yeah, for sure. This actually destroys a number of what were thought to be truths about herpes viruses, right?
Are you changing the textbook yet?
The text is at the printer, unfortunately.
Yeah.
I sent it to Jane, and I said, if you're in the next edition, you have to talk about this. Because she probably has some things wrong, right, according to this paper. So one of the nice things about this paper is the methodology that's used. I mean, the idea here is to look at the kinetics of mRNA production, both of the virus and the host, and translation of those mRNAs over time during an infection. And so they use three different methods. One is 4-thiouridine tagging of newly synthesized mRNAs. Because when you infect cell with a virus and you extract RNA, a lot of those RNAs have been there a while. They're not newly synthesized. So you add 4-thiouridine to your infected cells, and that will then be incorporated into newly synthesized mRNAs. And then you can attach a biotin molecule specifically to the thiouridine, and then you can purify those RNAs with streptavidin beads. So you basically can purify the newly transcribed RNAs. Didn't we do a paper recently that uses this?
We did a paper that used another method of labeling. So these are all non-radioactive labeling ways of labeling.
And this was that paper also was looking at newly synthesized RNA.
Right, and this actually provides a way to pull out, to isolate just the newly transcribed RNA.
Right.
Mm-hmm. I think the other one did too, or could if you use the biotin. So, yeah.
Oh, that was the splicing paper where polio inhibits splicing, I think.
Okay.
Maybe.
Yeah.
Anyway, so then you can sequence this newly transcribed RNA. You can do deep sequencing. So that's another technique that makes this possible. You can sequences of every RNA that you've pulled out there, and you know where they originate, and you can map them to either the viral or the host genome. So that's one technique. And the other is they want to know which RNAs are being translated. So they call this ribosome profiling. So basically, you take your infected cell, you break it open, and you pellet, or you centrifuge out the ribosomes because they're rather heavy and they will come out. And then you break them up. You break up the RNA. So now there's just a little piece of RNA attached to the ribosome where it's been moving along it. And then you can sequence that as well, and you can ask which mRNAs are actively translated in these cells, and you can, again, map them to the genome of the virus or the host cell.
This is just a level of information that it's amazing. You can see this.
Well, you know, in the old days, we looked at one protein and one messenger RNA, and as we will see, that can be misleading.
Yes.
Right? But that's all we could do. And I guess you would call this systems virology, right? Because you're looking at lots of things all at once.
If you were looking for a term for it, sure.
Yeah, not necessarily, but some people do call it that. When you do a lot of, when you collect a lot of data at once, some people call that systems virology. So they use these techniques to look at herpes virus infected cells. They take herpes virus, they infect human foreskin fibroblasts, and they take multiple time points, and they ask what kinds of RNAs are being made and what's being translated. So this paper has a lot of data in it.
Yes.
And it's actually displayed in very interesting ways, which I haven't seen before. Some of them very nice.
A couple of ways I had to look up.
Yeah.
Look up, and things I don't understand also.
Figure 1 is really nice. It's a diagram of these two methods, and then the scheme for how they isolate their fractions or samples at different time points.
All right. So the first experiment is simply to look in infected cells and see what's going on in terms of RNA synthesis and translation. And I really like Figure 2b, which is a graph, because it's something like I don't actually understand Figure 1a. I spent half hour looking at it.
2a, yeah.
I don't get it, and maybe
it took a while. I think I understand it.
I stared at it and looked up.
But I like B. Do you know?
But in A, if you look at those middle set of blocks along the bottom, they are darker than the left-hand set of blocks along the bottom. So that's the conclusion they draw. And then the rightmost of those middle blocks is a little bit lighter, describing how it drops off at later times in infection.
All right. Well, I prefer Part B, which is
nice graph.
XY
I
graph.
kind of could figure out what they were talking about with A, but I couldn't quite. I can't critically analyze panel A.
So in the graph, you see from uninfected to 8 hours with time points all along the way. They have total RNA, which they've gotten from their experiment, which goes up slightly. They're looking at percent virus sequences, or virus reads as it's called. Okay, so then they have the 4SU RNA. That's the newly synthesized RNA, which is it's put in red, but it goes up substantially. At about 6 or 7 hours post-infection, it's 80% viral. Okay,
right.
So it starts at 0 viral. Of course, uninfected goes up to 80%, and then the ribosomal profile, the blue curve, kind of parallels that red one. So you can see lots of translation of these RNAs in this.
Which is it's really interesting because the total RNA, they talk about the proportion of the total RNA that's viral, and it's way less than 80%. So the virus is successfully getting itself overrepresented in the translation machinery.
Exactly. That's the conclusion. I think this is a really nice way of... I mean, this has been known before that the virus shuts off the host, right, and replaces it with its own. But here they're using this technology to show
watch
this.
it happen, to see it. Yeah.
Yeah. Let's see. Increased transcriptional activity of cellular genes. So there's a little bit of increase of cellular genes was not matched by a respective increase in translation. Only 0.34% of the cell genes that go up showed an increased translational activity. So that's an interesting factoid there, which we will come back to. And in 77% of these genes, cell genes now, are not translated at all. So the cell genes that are going up, or whatever is there of cell, is not being translated or being translated at very low levels. Now, this sentence I really looked at about 10 times. They write, interestingly, 485 of 507 genes, 96%, with induced transcription, but no translation, were transcribed poorly or not at all. Okay, if they have induced transcription, how can they be transcribed not at all, right?
Yeah.
Now, I know what this comes to later, but it's just a funny construction anyway.
Yeah.
So the virus is taking over the host, but strangely, a lot of the host RNAs are not being translated. Okay. So basically, they have a lot of transcriptional activity revealed by this 4SU labeling. And when they map these RNAs, they see that they're extending into neighboring genes. They're not just nice transcriptional units of a single gene, but they have an example where they look at a couple of genes together. The transcripts are longer than they should be. And they have a lot of upstream, what they call upstream transcriptional activity, whereas basically the preceding gene seems to be transcribed and it goes all the way into the next gene, and that, of course, shouldn't happen. You should have transcription termination at the polyadenylation signal and then cleavage and polyadenylation and so forth. So something weird is going on. And some of them go as far as 100,000 nucleotides too far.
Yeah.
Which they call disruption of transcription termination or readout. And they quantified this.
And for those who might not have, well, because we're going to have read out and read in, you can think of read out as just being read through, way we usually have thought of it. Threw me for a while.
And read in is coming from upstream.
Reading into the next gene.
Yes. Reading into the next gene.
Yes.
So they have, of course, the ability to quantify this read out, read through. And
I guess read in and read out are both the consequences of read through.
Yes.
Because it's reading through the termination, and it's going to read out of the gene that was being transcribed and read into. I guess I see why they.
So at 7 to 8 hours, they have 9,600 genes on their data. Over 15% of the genes. This is a little complicated because then it introduces this other term. Let's just say it's a lot of the cellular genes that are experience this read through.
Yes.
All right. And again, this is specifically caused by virus infection, and how it does it is an interesting question. So this read through again goes thousands of nucleotides into neighboring genes, and only 4% of the genes. This is a better number. 403 of the 9,600 had less than 5% read out and read in. So read in is what again?
It's the transcription comes from before the normal start point.
Yeah, so read through is at the end, which is, as you said, it's basically a consequence of the same, not terminating, right?
Right.
Just depends whether it's upstream or downstream. Now, so only 4% of the genes showed less than 5% read out and read in, and this includes two genes, one of them GAPDH and another beta-actin, which people often use to study host mRNAs in herpes simplex infected cells. I found this wonderful in a way.
Yes.
And they say this may explain why the defect in termination has been missed so far. right? Because these are two genes which which escape for some reason this, which I'll call anti-termination. Isn't that cool?
It's just it was an unlucky choice by previous work. Yeah,
and this is the danger in just looking at using one cellular mRNA or gene to study a process, right? Because it could be the exception.
Right, but in the past, no blame on the researchers who did that, because
of course
in the past,
not,
you had no choice. You had to track something, and you would pick one thing that you thought was going to be representative. And of course, there's always been this caveat: we can only look at one gene, but now we can look at all of them and see what's really going on.
Another interesting conclusion is that they say for genes that have very low or no transcription in uninfected cells, when you infect them and you get this anti-termination, read-in probably exceeds the levels of endogenous transcripts, making you think that the gene is being induced, but it's not really. It's just you're getting read-through from another gene.
Right.
So it's not that particular gene isn't being turned on, but you get more of it because of the upstream read-through, and it looks like induction. And again, this is what people, if you do a simple experiment, you see an mRNA going up, it's induced, right? But it may not be.
Right. And you probe that mRNA level by whatever means. You could be doing it by radioactive old-fashioned blotting types of probing, or qPCR, choosing primers within the gene.
So in fact, this is important for many viruses. If your virus that you're studied happens to inhibit termination of transcription, you have to make sure you address this when you're looking for induction of genes, right? Many people will take a promoter of a gene that they think is induced and put a reporter next to it and show that that's turned on in a virus-infected cell. And that's okay, I guess, right? Because it's showing that the promoter is activated. But ribosome profiling, they found that this read-in for 99% of genes doesn't result in translatable transcripts. So all this read-through and so forth makes RNAs aren't translated.
Right.
All right, so that's an amazing finding in itself, but now they go on and they look at splicing. Because it's been believed that herpes virus infection results in inhibition of splicing of cellular messages. There's a viral protein called ICP27 that is thought to be a consequence of that.
And that also the fact that herpes simplex virus only has 4 viral genes that are spliced. So therefore, if the virus shuts down host splicing, it's not really going to hurt itself very much, anthropomorphizing the virus a little bit. Sorry, Jane. But that's the way it's been explained or rationalized.
So they can take their data and look for basically the ratios of introns to intact RNAs as a measure of splicing. And they see no increase in the intron to gene ratio throughout infection. In other words, there doesn't seem to be any inhibition of splicing according to their data. So splicing is generally not inhibited. Instead, disruption of termination of transcription and read-in is responsible for the observed increases in the levels of intron. So those are the intervening sequences in a pre-mRNA that are normally spliced out. When you get all this read-through, you get more intron.
Right. They saw a small increase, like a 2-fold increase in introns versus exons, but that certainly doesn't represent a global shutdown of splicing, and it's explained by this read-through that's happening.
Right.
Right. They do later say that ICP27 may be involved to some extent, and I think that you can't totally discount this 2-fold change in intron sequences. That's a really nice clear graph in that figure 6A.
Yep.
Just the levels of introns.
Right.
So they have an extensive discussion later trying to explain the 27 data, and I think you're right. It probably is playing some role. So basically, you're making these very large molecules spanning multiple genes, and they're getting spliced, but in different ways, which can contribute to getting higher levels of introns. Now, this is all hosts so far that we've looked at, right? All of the RNAs that they've examined as hosts. So they wanted to see if there's any effect on transcription termination in the viral genome. So they have all these data, right? When they purify RNAs from infected cells, data contains both viral and cellular RNA. So you can use a computer to sort them out, right?
Mm-hmm.
Right.
And now they look at the viral RNAs, and they basically try to correlate the expression with a known poly-A site, which is where transcription termination should occur. And they find no interference with transcriptional termination on the viral genome. Right? So the normal termination appears to occur, and the messages are the right size and end at the right place and so forth.
Right. So, for instance, if people are going to look at the paper, if you compare Figure 6, that's looking at a couple of host cell genes, and you can see the transcriptional activity between the genes, in other words, where the read-through is. And then you compare it to Figure 9, and you just don't see anything between the viral genes.
Yep. It's a nice way to put the data, right? Yeah.
And then they have the unfortunate way of saying that this shows that transcription termination is host-specific, and that threw me because I thought, wait, they didn't try different host cell types, so what they mean is that this transcription termination disruption only happens in the host, not for the viral RNAs.
I found the same thing, host-specific, right?
Right.
I don't know, what would be a better way to put it?
I just say happens only in the host.
Yeah.
Or affects only host genes.
Affects only host genes, that would be better.
Yeah.
Now there's a viral protein called VHS, and this is known viral host shutoff is what VHS stands for, and viruses that lack this gene have defects in shutting off host gene expression. So they asked whether such a virus lacking VHS was defective in interfering with transcription termination. And the answer is no. This deleted virus still can disrupt cellular transcription termination. Did you want to... we haven't gotten to Figure 8 yet, have we? No.
No.
Good.
Right, we skipped the splicing story. I mean, we talked a little bit about splicing and then went into the host-specific.
Right, but this novel splicing, we haven't really talked about.
Yeah.
Which could actually explain what's going on with the apparent, what was believed to be an inhibition of splicing.
Yeah, 44% of the induced splice junctions are novel.
Right. So you get these read-through products, and then they are spliced, but they're spliced wrong.
Right.
Because you have multiple genes put together, and it picks an exon and an intron and sticks them together, and it could be exons from different genes getting stuck together. So that's what they're working out there.
So they look at one region of the genome, chromosome 17 of the host, and they look at uninfected situation. That's the top part of this figure, and then the infected 7 to 8 hours situation. And then they do what's called a sashimi plot to look at the splicing. And I didn't really delve into quite how they did the sashimi plot, but they show with a red line what's the normal splicing pattern in kind of an unusual depiction. We have ways of doing it with carrots in the old-fashioned way. But then they show this blue splicing that's between the two genes, the MXRA7 and the JMJD6, just two specific genes in chromosome 17, and then another downstream splicing again in that same chromosomal region that's aberrant.
Right. So the sashimi plot is sashimi_plot is a function that's built into this software called Mixture of Isoforms software.
Yeah.
Mixture of Isoforms is given the acronym MISO.
Yeah. Perfect.
So in MISO, they needed a term for this software, among many other things. It deals with RNA-seq data and finds the splicing patterns in it. And this function produces a nice publishable graph of your data. And the plot that it produces shows the the raw data from your RNA-seq, get it? It's raw.
Yeah. That's
and the
sashimi,
splicing locations and the way it comes out is this this shape that they say. I think their explanation is a little convoluted, but they say it's somewhat reminiscent of a piece of sashimi lying on a plate. And also, they've already named the software miso and talking about raw data. So that's
do you get miso with your sashimi?
You get sashimi with your miso.
I'm sure you can work a title out of that, right?
Probably. So that's what's going on there. It gives you a plot of the data, and that's what figure 8C is, showing these little curved lines going through to
I think
the
it's
splices.
cool now. In 2 consecutive TWiVs, we've had reference to raw fish.
Raw fish.
Nice.
Well, and if you count steamer,
Yeah.
got a whole seafood thing going on here.
Yeah, seafood TWiV. Wow. All right, VHS is not required for disruption. Oh, and then they do an experiment where they infect. So far, we've used infectious herpes simplex virus. They infect with virus either inactivated with ultraviolet light or in the presence of cyclohexamide, a protein synthesis inhibitor, or phosphonoacetic acid, which I think is an inhibitor of DNA replication. Right, Kathy?
That's what they say.
They say
it only prevents
viral late gene. But I think from that other herpes paper we did recently, the double-stranded RNA thing from Ian Moore, I think they used also phosphonoacetic acid. And Rich Conda get all excited. That's a DNA synthesis inhibitor. Anyway, so cyclohexamide inhibits protein synthesis. Both UV inactivated virus and cyclohexamide treatment abolish this read-through. Okay, so you got to have some protein production. That's the cyclohexamide, and UV inactivation probably prevents proper transcripts and certainly translation as well. So you need some products to be made. However, phosphonoacetic acid treatment, which inhibits DNA synthesis and late transcripts, you still get the transcription termination. So it's not a late function.
Right.
And lastly, they have an ICP27 lacking virus, lacking the gene or a null mutant anyway. And they ask, do you need this for antagonism of transcription termination? Remember, the 27 was believed to be important for inhibition of splicing. But so they find it's not required for disrupting transcription termination. But there is some difference in the ratio of introns, right? It's slightly different compared to the wild type virus. So,
right.
So there may be some contribution to aberrant splicing.
So they looked at 3. They made 3 good guesses about what might be contributing, what might be causing this transcription termination inhibition, and none of them really panned out. I mean, what we have now is we know it is coming from the virus and it requires translation, and it's not a late gene.
Right.
But it's not ICP27, it's not VHS.
Makes sense that it's not a late gene, right? Because if you want to interfere, you want to do it earlier.
You want to do that early.
Yeah. Right, and it's not a tegument protein.
It's what the virus brings in, packaged in it, right? Those are all those proteins in there that the virus dumps into the cell, and one of them, what is it, VP16, is important for promoter activity of the immediate early genes, right?
So they've eliminated a lot of possibilities there, and I assume they have grad students and postdocs furiously testing the others.
It's an interesting problem, right?
Yeah.
Figure out how this is done, which will likely provide more insight into the termination process, although we do understand that quite a bit, but maybe this will be targeting some specific protein or proteins. It'll be interesting to find out.
Right, because it has a way to specifically inhibit host gene transcription termination, but not inhibit viral gene transcription termination, which is a pretty nifty trick.
So, yeah, that's very cool how it's selective. We don't know that as well.
So I just wanted to read one thing they do say about ICP27. Although it's not required for disruption of the transcription termination, it may contribute to the maintenance of efficient termination of viral transcripts.
Right.
Also they point out that expression of ICP27 is required for full late gene expression or full late gene transcription. Therefore, other viral genes may be involved.
Right.
But then, as you just pointed out, it doesn't seem like it's going to be a late thing
Probably
that
not,
you want to
yeah.
have happening.
No, because if you look at their time courses, right, it's happening earlier in infection. See that first graph, by 4 hours, there's already a lot of viral reads. So, I don't think it's a late.
Yeah. One thing that we kind of alluded to even just a minute ago is the fact that for a number of viruses, if you want to make sure that you're not misinterpreting your response to viral infection, you can't just look at the transcriptional activity. You need to know if there's some of this read-through that's reading into genes that you might think are upregulated.
Yeah, you have to look at it. You have to look globally, basically. You can't pick one or two genes either and just look at them.
Right.
So this is cool. The virus is basically, it shuts down translation by anti-terminating and making these long aberrant transcripts, which can't make protein. They don't get spliced properly. So as our reader Pete said, it's a new way of shutting off the host, right? It's a novel mechanism. In fact, they say here, disruption of host transcription termination has not been reported before for any pathogen. That's pretty cool.
And the fact that these things seem to get hung up in the nucleus to some extent too. I get that?
Yes, that's right. These long transcripts get hung up in the nucleus, right?
So I talked to a colleague about it, and what he pointed out, what he would like to have seen is some analysis looking at some of these precursor RNAs, these longer things, something like a northern blot, something to show the intact messenger RNAs or these long read-through things.
Yeah.
That might have been a helpful, convincing piece of data.
I try to get people to do northerns, and they look at me funny.
Yeah.
Why do you need to do that? Why don't you just sequence it or do PCR?
Yeah.
So this is pretty cool, and it leads to a lot of other experiments as well. No inhibition of splicing, no general inhibition of splicing. Right. This is important because now, people, as I say, 25 years ago, people said it inhibits splicing using, individual genes and in vitro systems, and it doesn't tell you the whole story.
Yeah.
You need to look at newly synthesized RNAs globally.
Which now we can do.
Right. And I like the last paragraph. So I immediately said we have to do this for our virus because I think this would be really interesting to know, what RNAs are made and is there any effect and so forth. As they say in the last paragraphs, we would like to note that our approach is not only applicable to any virus or pathogen, but could also be extended in vivo. Owing to its outstanding temporal resolution, it's well suited for pathogens with short replication cycles. That's great.
Yep.
It's really nice. So that's a good paper. Thank you, Pete, for pointing that out. And I hope our listeners don't get discouraged. It's a little complicated, but, sometimes we do these, I don't know, what would we say, molecular biology heavy papers, but I think it's important. It's part of virology. Without this, we wouldn't have vaccines. Well,
right. And this, could be pretty much paradigm shifting.
Absolutely. I would love to.
And downstream of this, I mean, there's neat stuff going on here that we previously misunderstood or had no idea was going on.
Did you make a pun downstream?
Oh yes, I guess I did. So reading through this paper.
Oh, that's a great title. Reading through this paper.
Yeah.
So many good titles.
Yeah, there's a lot that could come out of this.
All right. Let's do some email. Start with you, Alan.
Okay. Actually, these first...
Ah, that's right. I meant to say
it's a
this.
cluster of them.
These are 1, 2, 3... Let's not read them. I just wanted to... I just wanted to point out what they're about. 1, 2, 3, 4... Four
letters, all about the same paper.
Yeah, so this was
from Jacob, Fernando, Cedric, and Alice.
Right.
So this is a paper published in Science. Long-term measles-induced immunomodulation increases overall childhood infectious disease mortality. So this is a perfect paper for TWiV. Basically says that, measles virus infects immune cells, and so it immunosuppresses you. They looked at data available on the incidence of infections with or without measles immunization, and it's quite clear that if you prevent measles, you prevent many other infectious diseases as well.
And this is actually, that part has been known for decades.
Mm-hmm.
There's this observation from public health that you go and when the measles vaccine came out and when it was more widely deployed, everywhere that it was used saw a sustained increase in all sorts of child mortality and child illness measures. And everybody has just kind of shrugged their shoulders and said, well, overall health improved, and that was part of the measles campaign. And so this actually comes up with a mechanism.
Yeah. So we did this on TWiV.
Okay.
So Michael Schmidt basically grabbed it a couple weeks ago. He says, I want to do this. So we did it there, and I'll put a link to that episode.
Okay, so Jacob, Fernando, Cedric, Alice, and everybody else who was interested in this paper should go over and listen to it.
Check it out. It's perfect for TWiV, right? It's something really relevant, and so I appreciate that, but go check it out on TWiV. The last person, Alice, wrote, if you discuss it, you should get a pediatric ID physician to join you. And then she says, in general, I would be so excited if you could find an ID doc to join your discussions.
Sure.
I'd love to have, let's say, a monthly basis to have an ID doc, right? I think that would be good. Because we don't know, like today's paper, an ID doc would not really want to participate in the discussion, right? So I'm kind of searching for someone to join us on a monthly basis. I know you guys are busy, you ID physicians, but this is important. Yeah,
because the show really has the core hosts and then has a rotating set of other folks who we have on pretty frequently. I mean, we were just talking about Matt Freeman commenting on the MERS paper. We've had him on several times. We've had Ian Lipkin on several times. So, yeah, having an ID doc on call, so to speak.
On call, right. All right, now you can start the next one, which is Anthony. Okay,
so Anthony writes, quoting from that article, What We're Afraid to Say About Ebola, and this is the notorious Osterholm editorial that whipped up public fear in the Times. And quoting, the second possibility is one that virologists are loath to discuss openly, but are definitely considering in private, that an Ebola virus could mutate to become transmissible through the air, which is something we debunked openly here on the show at the time. And then Anthony's then citing another article. This is from SIDRAP. Change in pattern of H5N2 spread raises concern. Another infectious disease expert, Michael T. Osterholm, said that changing transmission picture doesn't necessarily mean the virus has mutated. He's director of the University of Minnesota Center for, which publishes SIDRAP. Quoting Osterholm, there's been evidence of transmission like this before in the Netherlands, so I'm not sure this necessarily means that, he said. It could, but we need the isolates tested to find this out. It could be just acting like any other highly pathogenic avian flu virus we see with wind-driven virus transmission. It just hasn't been stopped by the current level of biosecurity. And links to that. And then Anthony says, I'm not sure what Michael Osterholm means by evidence of transmission like this before in the Netherlands. The cited study found virus material in surrounding air, but did not detect any infectious virus, perhaps due to the sampling procedure. My understanding is that influenza is sensitive to drying and to UV. It was distressing that the press was being told that wild birds were the cause of the current poultry epidemic, as there was no evidence for that. If investigators are now in a check every and any possibility mindset, that certainly is an improvement. Is that what's going on, or are platitudes being replaced by wild speculations? Ebola did not, quote, become transmissible through the air, end quote. Is it any more likely that bird flu is blowing in the wind?
So this Kathy, relates to the fact that I heard Michael Osterholm quoted on NPR last Thursday, and then I told Vincent about it in person. And the quote is, "Influenza viruses have thought in the past to be transmitted by birds to birds in close contact, and that it was only through that kind of transmission that we need to be concerned. Now we surely have a very dynamic situation in the Midwest. It's also a situation where we can no longer assume it's just migratory birds." And I just don't think that's how we have thought about it in the past. For instance, a review article from 1995 talks about transmission of the virus avian influenza from wild birds. In the vast majority of influenza outbreaks occurring in domestic poultry, particularly turkeys, spread from wild birds appears to be the most likely mechanism of primary infection, the evidence being weighty, though mainly circumstantial. So, to say that it's doing things we've never seen it do before is just not right.
I agree that this has been suggested for a long time, right? Maybe we didn't prove it, but that's what this PLOS One paper is trying to look at. Airborne virus, although they didn't isolate it, actually infectious virus, so there's a little bit of a question there. But I agree. I just don't think this is anything new. It may happen on a larger scale because everything is getting bigger. So,
and the poultry farming industry is, how should I say this? It's a messy business.
Certainly, yeah.
A lot of chicken farming I know is structured, and it's almost a scam what they do to the chicken farmers. They get them to invest huge amounts of money and put up these huge, massive facilities to raise huge flocks, and when that farmer goes bankrupt, they go to the guy down the road and have him repeat the process. So there's a lot of movement of poultry from place to place. It's very poorly documented. I don't think we've rigorously excluded the possibility that this is spreading just from farm to farm through poultry movement. And certainly wild birds are another part of the equation. Whether that's the whole story or not, I don't know. But these are not biosecure facilities, if you will.
Right. Not in any way, shape, or form.
These are big, huge metal buildings sitting out there in the plains, and if you want to slap the free range label on it, then you've got a little door in the side that's open, which means the chickens don't really go out, but the sparrows or whatever go flying in. I really don't think that it's appropriate to be speculating about avian flu blowing in the wind. That's just not. I don't think that's a mechanism we need to invoke yet. We should look at more obvious possibilities first.
And I don't know if you noticed, Kathy, outside the building at Cal State last week, there was a PETA booth,
right?
And the theme was how chickens are raised so crowded. They had a big picture of all the crowded chickens, and you're supposed to go inside and sense what it was like to be a chicken in one of these facilities. I didn't go in, but I felt like going up and I say, "Well, these are a problem for influenza. Did you know that?" They did not. All right. Kathy, can you take Ken's email?
Ken writes, "Dear TWiV, thank you for your fantastic job discussing my article in episode 313, with viruses like these, who needs enemies. I first found out about TWiV when a colleague here at NYU told me that you guys discussed this paper from my lab, and I've been listening ever since. I am particularly delighted to see your enthusiastic coverage of the virome and the unappreciated effects viruses have on the host, which brings me to the reason why I'm writing to you. Not too long after I started listening to TWiV, I was asked to write a review article on the virome for the journal Immunity. In one of your episodes, you discussed how the immune system doesn't make sense without viruses, a variation on Theodosius Dobzhansky's famous quote, "Nothing in biology makes sense except in the light of evolution." Although I think you guys should still learn what a TH17 cell is, I took inspiration from this and other discussions that arise during your podcast, which I believe comes through in the article I wrote. I've attached the uncorrected proofs of the review article, which I hope you will find interesting as virome enthusiasts. The emphasis is on the role of mammalian viruses on complex diseases and the relationship between the virome and the microbiome. Most importantly, I acknowledge TWiV in the acknowledgement section. The service that you provide the scientific community and lay public is appreciated. The article is scheduled to come out in the May 19th issue. Sincerely, Ken. And then he adds a PS about not discussing it until it's published, but it has now been published.
No, we're so far behind on our letters, you could write about stuff that you're just starting to work on.
And is this the first acknowledgement in a
Yeah,
published paper to TWiV?
Absolutely. I've been waiting, for someone to...
Cool.
I'm surprised the journal let it get in there, too.
Yeah.
I think,
Yeah.
what is this TWiV thing? That's pretty cool. Thank you.
I wonder what our show's H-index is.
So did we not know what TH17 cells are?
I think we...
Screwed it up?
yeah, and then we were corrected the next week or two.
Yeah, I think we were confusing IL-17 and IL-17 knockouts with TH17 knockouts, right? Yeah, that's right.
That sounds like us.
Yeah, well, yeah, it's okay. You got autoimmunity, right?
Right. Autoimmunity.
Autoimmunity. Or
Autoimmunity. Just have me say it.
Autoimmunity.
Yes, exactly. Kathy, take it away.
What is it?
Autoimmunity.
I think it should be auto and have a car as the...
But this is the whole thing.
Yeah,
It's
I
just
know.
audio.
I think
It's
they should get a co-host named Otto.
In your talk last week, Vincent, I actually pulled it up and took a screenshot of it on my iPhone. I was going to show you later, and I forgot.
Autoimmunity?
Autoimmunity.
Autoimmunity? How
There
can
you
you
go.
have a podcast where it's so bloody hard to pronounce?
It's not hard at all. Autoimmunity.
Look,
I say it every time.
I have a PhD. It's very hard for me.
It's not a PhD in pronunciation.
I'm just kidding. I just think you should make an easy to pronounce podcast.
And remember, their organization or whatever is immunity. So they did a couple of things so that they wouldn't always come up with the immunity hits.
Yeah.
But you can find them.
I think I would have named it differently, but it's not my podcast. When I do make an immunology podcast, it's not going to be difficult to pronounce. I know it's not really difficult. I'm just being a pain. Next one is from...
But when you make an immunology podcast, it'll be difficult to correct.
Yeah, well, I won't be doing all the talking, It's
Right.
going to be somebody else, and I'll just ask questions. Thomas writes, Dear TWiV Amici, during
Excellent.
the listener email segment in TWiV 335, Justin wrote in suggesting that we should consider doubling the grad student stipend, which would limit the number of graduate students that graduate programs and PIs could afford to train. With this proposal, Justin acknowledges a major issue in science, the significantly greater pool of trained PhD scientists compared to available jobs. Alan rebutted with almost the opposite, a training model that is similar to that of medical training in the U.S. and requires the student to pay tuition and all living expenses. Full disclosure, I'm a grad student at the University of Michigan working on preparing a manuscript, and originally was not going to write in on this matter, but was convinced to do so by Kathy after her and I discussed the points brought up during the aforementioned segment. I'm also Canadian, and I'm familiar with the Canadian system of graduate training. Finally, I'm an MD PhD candidate that is not part of an NIH-funded MSTP program and paying for my MD training, so I'm quite passionate about any discussions concerning funding of physician or scientist training programs. With that, I would like to leave you with a few points to consider and possibly discuss. One, decreasing the number of PhDs seeking jobs after graduate school should allow for a situation similar to that in medicine, where graduates are almost guaranteed employment. With regards to the repayment of graduate or professional school debt, Alan assured us that physicians are able to eventually pay off their medical school debt and live comfortably. Physicians are, however, also guaranteed, on average, much more in pay than most scientists can ever dream of earning. Considering that the cost of tuition and living expenses for both medical students and graduate students are comparable, this begs the question whether such a model could ever be fair or successful in research.
I would quibble with your pay scale, and I would be interested to hear from you after you receive the MD and do your residency and are out working in the field.
So you don't think it's as high as he's saying?
Physicians do make good money, and they are highly employable, but scientists who actually manage to land a job and get a grant can earn a comparable income to many physicians, and many physicians who are employed, particularly in fields like, general practice or pediatrics, are not particularly well paid.
Yeah, I would agree depending on... So, in medical schools in New York City, yeah, I would agree that you can get paid pretty well. An undergraduate institution in Bozeman, I don't think as well.
Right.
So
But your cost of living is lower, too.
Yeah, the cost of living still does not outweigh the difference in salary that a physician and a scientist would make. So,
But it also depends on which branch of which specialty of physician you're talking about.
I agree, but I would think that even the less well-paid physician salaries are going to be higher than scientist salaries.
I've known pediatricians who make less than postdocs.
Wow.
So are these data available?
Data on physician pay is available, and you'll find that it varies radically by specialty.
And I think the pay for scientists, professors is also available.
Yeah,
too,
pay
right?
for funded scientists on NIH grants should also be available. I don't know if anybody's actually put these two together, but I would quibble with the blanket assumption that MDs are automatically going to make more than PhDs if the PhDs actually manage to land jobs and funding, which is the big if.
Yeah. Right. But I would guess that if you looked at the aggregate average of scientists and considered all universities and didn't qualify it with those that managed to land an NIH grant, and you compared that to the aggregate of all physicians, that physicians are going to make more.
But of course they are, but that's a very different question, because the issue there is the difficulty of getting a funded position in science.
Well, but it goes back to what the writer is saying, in that if there are guaranteed jobs at the end, which there are for physicians, then you would be willing to take that risk and take on that debt. But that's not the case.
And if there were guaranteed funded jobs for PhDs at the end, I can virtually guarantee that the aggregate pay for PhDs would be completely different from what it is now.
I'm not so sure that's true, but we can go on, continue reading.
Continuing the letter.
Right. All right, 2, just as increasing the pay of graduate students is a circuitous route to a more selective admissions process and decreasing the number of trainees, so is eliminating the pay. We should just accept fewer students. Moreover, one can imagine a situation in which graduate students are to be paid for by the trainee, thereby providing institutions with not only free labor but also a financial income. Unless strict regulations also accompany such a model, I would be concerned that some institutions may take advantage of this situation, be less selective, and irresponsibly admit many more applicants.
A valid point.
Yeah, I would agree with that. Although, here, trainee PIs pay for students after the first year, but the first year is still paid by the university, and that does not allow for an increase in the number of students that are accepted here.
Right. And I was making a radical opposite proposal just to kind of be contrary. But if you do have some kind of pay, even if it's a very low pay for the trainees, for the PhD candidates, that would also give the NIH the leverage to enforce a quota on them. Because they could say something like, we will only allow you to have one graduate student per R01, for example.
Yeah.
Which would drastically cut the number of PhD, or one graduate student or one postdoc. You could even take it to that level. The point was, we need to drastically cut the number of people entering the field to train as scientists, because the jobs they're implicitly being promised don't exist.
Yeah, I think so. What we do here is we require that you have 4 future years of NIH funding to have a student. If you have 3 or 2, you can't take a new student until you get 4 at least. Which the idea is that would be enough to complete a student. And I'm just worried that I think you're right, but I think we need to have more jobs for some. We need to have more science in general, because science is what drives the world, and we don't have enough.
That'd be great.
We don't have enough.
That would be great. But
I'd hate to cut back in, let's say, 50% of PhD trainees, and then all of a sudden, in 5 years we realize we need more scientists.
Not going to be a problem.
We can ramp it up really quickly.
There is no such thing as a labor shortage. That is absolutely not going to be a problem. If you find, let's say you cut the number of trainees by an order of magnitude. If you do that and you find 10 years down the line that suddenly Congress has decided and the taxpayers have agreed that we need to quintuple the NIH budget, you will have absolutely no difficulty whatsoever getting trainees into a program for those jobs. And
but then there's still going to be a 5 to 7 year delay before they're able to.
You will have more than enough time to get them trained up for those positions, because you're not going to have, contrary to legend, you're not going to have a huge population of scientists instantly retiring tomorrow. So we have such an oversupply now, and these types of labor problems can always be solved much faster than the people claiming them ever will admit.
So you think that people, for instance, who have
Also,
left
that's
science
not
or
gonna
will
happen.
have left science in the interim would then come back?
Oh, sure. If somebody offered to set me up a lab, yeah.
Okay.
I mean, there's this vast pool out there that could do that, and also this is an entirely fantastical
of course, I understand.
thing.
Speculation,
It's not going
right.
to happen that this type of funding bolus suddenly shows up and sticks around. So what we need to focus on is building a model that is sustainable, which is going to require, my fundamental point was we have to unlink the training from the staffing.
Yeah, right.
And one way to do that would be to stop paying the people who are training. But another way to do that would be to just cut down on the number of them who are in that program. But the idea of doubling the pay for trainees doesn't come anywhere into it.
All right, one more point, number 3. Finally, how do you all feel about a model where candidacy in a PhD program can only be achieved after completing a master's degree or transferring from a master's program due to outstanding performance and recommendations? Such a model would require students to pay for their master's education, with scholarships for those unable to financially afford, but academically apt to do so, of course, but then see a fraction of those students transition into a PhD candidate position if they felt inclined and were competitive enough of a candidate to do so. This model is already in place at several institutions around the world. I'd be interested to hear what you all think about this model.
Well, at least it takes care of one aspect of the whole thing for me, which is if people are having to pay their way, that it would really cut out a number of people who financially just cannot afford to do that. But he's built in the some kind of scholarship or something.
So I've seen enough master's programs to be highly skeptical of their value, right? You know, we have a number here, not at the medical school, but downtown, and I think they are largely a revenue-generating program.
Yes.
And so unless you made master's programs that would be rigorous and would provide a useful way of evaluating whether someone was appropriate for a PhD program, I don't think it would be worthwhile.
And if you did that, you would have the current system. I mean, I have a couple of pieces of paper on my wall that say Master of Arts and Master of Philosophy, and the only purpose of those degrees was to qualify for the PhD candidacy.
Yeah.
So we started or kind of revamped our master's program, I don't know, and it probably came into place 5 or so years ago. And it was judged in several other departments or seen as a way of generating revenue. And we looked at it pretty carefully and figured out that it wasn't really going to be very revenue generating, but that it could serve a purpose for people who wanted to increase their academic credentials for perhaps applying to graduate school or medical school, or just wanted to have a master's degree because that would set them up better in industry or something like that. And I think a master's program could be made rigorous. In fact, it could be made even more rigorous than ours is. For example, we allow students to take our graduate level classes, or they can take upper division / dual level courses. If we required them only to take our graduate level classes, then it would be a bit more rigorous, and it could serve as a way to really identify the students who were qualified to go on for a PhD.
I think, though, that these are all. I mean, that sounds like a great idea to make the master's program more rigorous, and I would favor that for its own sake. But using that as an additional barrier to entry, I don't think is useful. because it's not it's not really the issue that you need to get more qualified candidates into the program. It's not that we're that we're producing lots and lots of incompetent PhDs, I don't think. The issue is that we're just producing too darn many of them for the amount of science that we can afford. So given that, what needs to happen is some kind of an implementation that will force the universities, because it's not the students that are the problem, it's the universities that rely on the students as part of the research structure, as part of the staffing. We need to force the universities to go to a different staffing model, and something that strictly limits the number of graduate students that any given program can have is, I think, the only reasonable mechanism to do that. Because otherwise there'd still be this incentive for programs to bring in too many students and exploit them.
Yeah, I sort of agree that making a rigorous master's program is sort of like duplicating what we already have in place here in the first few years. And
so then, then that kind of a student wouldn't maybe take as long to get the PhD, and it would serve to discriminate for students perhaps that come in, and rather than getting all the way through 5 years of a PhD and finding out that they're not going to go on to a job in academia, they could do it after 2 or 3 years and go to a job in industry, or be a more highly qualified teacher in secondary schools or something like that.
I mean, the reason I like it in some ways, because I see too many PhD students come here right out of college, and after a year they decide it's not for them or they don't like it, and that's a waste for everybody.
Mm-hmm.
Right.
So having a master's program would either get those individuals earlier, or they would be discouraged because they had to pay for it on their own, and if they're wishy-washy to begin with, that might make the difference. But to get all the programs in place that are rigorous, it's not going to happen immediately, right?
Right. But as Thomas says, it does happen in some places around the world now, so it would be interesting to take a look at those and see how the supply and demand of
the problem, Kathy, is that if say our department said, all right, you need a master's to apply, immediately everyone's going to apply everywhere to Michigan, and they'll get into Michigan, no problem, and they'll all go there. Right? So nobody will make that first move.
Yeah, I think there's some of the stuff coming out of the FOBGAPT meeting that may have some more unified actions on the part of universities. So that's that meeting that was here at Michigan a couple weeks ago, and I haven't seen any report out from it yet, but you can go look at things online about it.
Well, NIH could make a stance here by not funding training grants anymore, or cutting them substantially. I mean, I know they have cut them substantially over the years, but they could say, all right, we want to put this money into staff positions or something, so they could have an effect at that level.
NIH is the only operation that can change this, because as long as they're running the plantation, that's ultimately, this is all about grant-funded science.
Yeah.
And all the graduate programs that are cranking out all these PhDs are doing it to staff the grant-funded science that they're doing. If the NIH says, hey folks, here are the new rules, you can't have so many graduate students, then instantly all the programs will, by whatever mechanism, they may come up with many different mechanisms, and that's fine, but they will all have mechanisms for drastically reducing the number of PhD candidates they admit because they can't use them.
Well, there were NIH people at this meeting, to my understanding, and there's also other funding agencies like NSF and USDA that would figure into this. So it's not only the NIH, but it also is going to have some buy-in from the university side, and that's what I got the impression there might be coming from the kind of meeting that was here.
Right. Yeah, I mean, the NIH can't just unilaterally act because the universities have a powerful lobby, but they are the 800-pound gorilla in funding our field. Yes, there are other funding sources, but I'm certain that if the NIH set these ground rules, the other agencies would either follow or it wouldn't matter.
Right.
Because they just don't have the clout to affect it that much. So yeah, this ultimately has to come from Bethesda, and there are, I think, fairly simple but drastic measures that they could take that I don't know if they have the political will to do it, but your suggestion, Kathy, is that maybe coming out of this meeting that they will.
Yeah, my hope is that it wasn't just another meeting to talk about what the problem was, but that they did have a lot of kinds of solutions, and then there's scheduled to be another meeting next year in a more formal venue or planning called the Michigan meeting. So the Michigan meetings are something that's been going on for years, and so this was an ad hoc Michigan meeting, as it were, and next year there'll be another one. So again, was it just another meeting to talk about what the problems are? I don't think so.
Good.
But time will tell.
All right, so continuing with Thomas, so much for writing a short letter and getting back to work. Thanks, Kathy.
Yeah, he saw me the day after that TWiV, and he was just fit to be tied. And I said, well, just write into TWiV, into TWiV. And he said, I don't have time. And I said, you have time. Write the letter.
It's an important topic, and we can talk for a long time about it.
As we have.
Thank you for all of the time and effort you devote in order to produce this podcast. It has been a significant influence in my early career, as it seems to be for so many other listeners. Let's read one more, so we can end on a nice
on a positive note.
Yes, we are around to
an optimistic note.
Alan.
Yes. Daniel writes, "Hello all. I started listening to your podcast at TWiV number 6, and I'm still loving the cast and all the spin-offs. I've listened to every episode of all 4 casts at least once, and my only wish is that you could make more or do them more frequently. I've never listened to one of your podcasts and thought it was too long. I love the science, I love the banter, I love the puns, I love creating new TWiV star disciples, and I have about an 85% success rate if I can get them to listen to one episode. Other than the customary sycophantry, I just wrote in for a couple of listener picks. These are all along the lines of don't provide links to anything you don't want people to reshare. And he provides this link to, let's see, an article in The Guardian talking about false equivalence and this notion, which is quite accurate, I think, that the media has done a general disservice on science reporting by pretending that there are 2 sides to every story. And sends a couple of other, let's see, what are these other links?
Facebook pages you need to stop sharing.
Top 10 Facebook pages you need to stop sharing from. And
part 1 and part 2, it looks like. Yeah.
Yes. So this is a 2-part piece on debunking various sites.
Yeah, like Mercola and anti-vax stuff,
Yes.
The do not link stuff, right?
Yes.
Yeah, it's stuff you shouldn't share, but too many people do.
People do.
And I'm assuming that to complete his record, that Dan, after starting at number 6 of TWiV, went back and listened to 1 through 5.
I guess.
So.
Yeah.
Yes, he said he's listened to every episode of all 4.
That's
right, so
very
he must
impressive.
have. Yes, that is impressive.
Yes. All right. And P.S. It's 77 Fahrenheit, 23 C, and partly cloudy in Socorro, New Mexico.
Socorro.
Did we do weather?
Here, no, today we did not. 75 degrees,
Ooh.
partly, mostly sunny.
Okay. It's 82 and mostly cloudy here. Well, they're claiming mostly cloudy at Bradley Airport, but I've got what looks like mostly sunny out my window just a few miles north of there.
Mm-hmm.
It's 27 C here. It's overcast, though the sun is pretty warm.
Yeah, it's been summer. I love it.
We had on TWiM, we had a guy. Is it TWiM? TWiP. There's a guy from Hawaii who listens to TWiP. He sent us his weather, and then he had something to do with the effect of volcanoes on visibility.
Oh.
There's a number. Let me get that up. I have it right here. TWiP 90. That was yesterday. We had an interesting one here on TWiV not too long ago. But here we go. This is from, sorry about this here. Matthew, Federico, where is it? Alan, come on. No, not you. This guy's name was Alan, yeah. He's in Hawaii.
I'm definitely not in Hawaii.
Just thinking about this, though, it's
There
kind of
it
funny
is.
because
Here
people
it is.
listening to this episode, who hate the weather, probably thought, "Oh, wow, they didn't do the
They
weather.
got through a whole
escaped."
episode without doing the weather. Arr!
Yeah, sometimes I do things on the road and I completely forget the weather, and then people are very upset about that. Why didn't you do the weather? I was waiting for it. Sometimes I forget. Okay, here it is. The weather in Kona today is 77, partially cloudy with VOG, volcanic fog, so thick I cannot see the horizon, though it's there somewhere. VOG, volcanic fog is VOG. There you go. That was a new one.
It is.
All right, that's it for our email. Actually, we only have a couple left, which is amazing. We've almost caught up. Can you believe it? So let's do some picks. Alan, what do you have?
I have a hoax that was just recently revealed by a science journalist named John Bohannon, who writes for a number of outlets, including Science. Bohannon did a thing a little while ago where he sent a fake paper to a bunch of open access journals and found that a number of them accepted it uncritically.
I remember that.
Basically revealing what we knew was the case with the scam journals. We're not talking about PLOS here. We're talking about the whole cottage industry of journals that just churn out crud to get the page charges. Anyway, he was apparently contacted by some filmmakers who were doing a documentary about how much BS there is regarding nutrition. And as part of this hoax/PR stunt, he worked with them to set up a fake clinical study, which they got a dozen people to suggested changes in their diet, have a chocolate bar with your meal versus the control group, which was told not to, or was not told anything of that sort. And they tortured the statistics until they got the right p-value, and then submitted it to a scam journal that they knew would publish it without review. And
he sent it to a number of journals, actually.
He sent it to a number of journals.
Yes.
From his list that he maintains of scam journals, and these that are PubMed indexed, but they essentially will print whatever you send them. And so he got it published 2 weeks later, without any kind of peer review apparently, and then cooked up a press release and sent it out on this very typical PR type of stuff that you see with these sorts of poorly designed nutrition studies. So he's really just doing something that is done routinely all the time and that produces exactly the type of story that this produced, which is that chocolate accelerates weight loss. Okay, and it got picked up by a bunch of news outlets, and essentially they reprinted the press release in multiple languages, didn't do any kind of fact checking, and he describes the entire situation here in this this blog post that he did, or this article that he did.
And he does a really nice job of explaining the statistics of it, because they found something that was statistically significant. And the gist of it is, if you look at enough things, weight, cholesterol, sodium, blood protein levels, etc., you're going to find something that you can claim as statistically significant. So they knew they would find something.
Even calculates the odds that they would find something reportable that was statistically significant, and it was better than even odds that they would come up with something. Yeah.
Right. It's really well done.
Yes.
Yeah, it's great. It's excellent. So he had done some other things on open access previously.
Yes.
Right.
Yeah, so he has shown a spotlight on this particular cesspool before, and this was, I think this is just a work of art. I was very pleased. I mean, not pleased with the situation.
Yeah, of course. So he is a critic of science publishing, basically, right? Good.
Well, he's a science journalist, but he's a critic of the current state of much of what passes for science journalism. And of course, people have been hammering this point home for years, but it never seems to get anything done. So now he's essentially warning the audience of these types of outlets that what you're getting is a load of BS.
You need to listen to TWiV and autoimmunity.
Of course.
Audioimmunity.
There you go.
Yeah. And all the others. Kathy, what do you have?
I picked a very short video that is this whatever it is called again, New Horizons spacecraft that's beyond the orbit of Neptune, and as the astronomy picture of the day says, closing in fast on the solar system's most famous unexplored world, because they don't know what to call it because it's not a planet anymore, Pluto. And it's a 4-second YouTube video with 13 frames taken over 6 or 7 days. And it says, although blurry, the images rival even the best images of Pluto taken from Earth. And it's going to pass Pluto on July 14th. So I don't know how close it's going to get, if it's going to get any closer than this, but we may have some more pictures yet to come. But I just thought it's a cute little short video, and you can see Pluto and its largest moon, Charon.
Yeah.
Sure is short. Yeah. That's really the best we have, huh?
It says it rivals the best pictures taken from Earth. So
there's been very little study of Pluto.
Yeah.
So far out there.
I didn't know that Astronomy Picture of the Day is brought to you in part by Michigan Tech.
Yes.
Yeah, that way you like it because it's a Michigan thing?
No, I just like it.
Yeah. My pick is the ASM Agar or Agar Art Contest.
Thought it was Auger.
Auger.
Some people say Agar. Some people who are in the know, not me.
They're in a different country, I think.
Agar. You say aluminium?
I think that might be those people.
Anyway, this is a contest to create a piece of agar art, and you can enter this contest. The links are there at the website, which I'll put. You'll have to submit everything by September 3rd, and so you have time for these things to grow up. I'm sure you have seen various agar arts all over the internet doing cool things. We've shown them here before. So this is going to be an official contest, and you're going to get... first place will be $85 credit at Michelle Banks' store on Etsy. She makes this cool microbe art. Mm-hmm. And then first, second, and third place, and People's Choice winners will receive their choice of an ASM book from the list provided here. Let's see if it includes principles, huh? These are probably all principles of something, no principles of virology, but there's principles of microbial diversity, which I think is one of the books we advertise. To Catch a Virus is on this list.
Oh, yeah.
Microbes and Evolution, The World That Darwin Never Saw. That's a good one. Antibodies for Infectious Disease. So you should check it out. If you were in a lab, and or actually you could do this in your garage if you're properly set up, but don't tell you. Sure. Don't say that I said it.
You could do it in your kitchen if you're using yeast.
Yeah.
Right.
So check it out. I'm one of the judges, so I will be checking it out along with Michelle Banks, the artist, and several others. Okay. And
there are some guidelines for biosafety so that you should be safe as you make your agar plate art.
Yeah, you shouldn't use Bacillus anthracis.
No.
Even if the Pentagon sends us to you special delivery.
Right. We have a listener pick from, we had a couple already from, I forgot who, sorry. Now we have a pick from Ken, who writes, my son's fiancée pointed me to a new citizen science project she is involved with on Kickstarter. It is called Bird Box Science, and is a module of sensors you install in a bird box to gather nesting data, such as temperature, entries, exits, and video. Then the data are uploaded to a crowdsourced database for anyone from researchers to educators to individuals to view and use.
Oops. Bird Box Science funding was canceled.
Too bad. I guess I didn't give it the
canceled on May 20th. Yeah, we didn't get to it soon enough, I guess.
Well, you can still read about Ken. It. It's still got nice diagrams. Anyway, he wanted to check it out, but sorry. Anyway, Ken's in Snohomish, Washington, where they
got 2,000 out of its $40,000 goal.
Mm.
Well, yeah, you got to set it low, right, and then you go above it. All right, that's it for our listener picks. That's it for TWiV 339. You can find this and all the others at twiv.tv and also on iTunes or using your favorite podcatcher. And I wonder how many other people have listened to every episode besides Daniel. Was that Daniel? Yeah. Wasn't that great? He loves everything about it.
Yeah.
Not too long, not too short, no bad jokes. creating new tweed disciples.
Wait, I don't think he said there weren't bad jokes. He just said he enjoys the
Hey.
puns.
Right, he just enjoys them. Right.
I love the puns. That means I love bad jokes.
Yeah.
That's right. That's my interpretation. How many people have listened to all of them besides us? And send us your questions and comments. TWiV@TWiV.tv. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was fun.
Alan Dove is at turbidplaque.com. He's also Alan Dove on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.